Metrix. Clemens. He'll be telling us about extracellular vesicles and their characterization using nanoparticle tracking analysis.
Good afternoon, ladies and gentlemen. I bring you some updates about NTA technology applied in the field for bio-nanoparticles such as exosomes and liposomes. Particle Metrix is a German-based company. We not only distribute particle analyzers such as NTA and DLS, we also develop, manufacture, produce in Germany, as well as quality assurance is done in Germany, and then we distribute and ship worldwide. So an NTA technique, you're mostly familiar with it, but for the brevity of it, it's a quite simple and straightforward technique. You inject your sample, you visualize it. It's actually a microscope, an ultra microscope, and then you can continue or start measuring. It's rather quick, rather straightforward, and basically it's not a very new technique. It's an old technique. Back 100 years ago, an Hungarian-Austrian researcher named Zsigmondy introduced the concept of ultra microscopy into the field. Basically, you have a laser, or at that time it was a strong light source, and perpendicular to it you have a microscope, so you magnify what you see, and what you see is not the particles, actually it's a reflectance pattern of particles, so you track the Brownian motion of it, and you analyze the Brownian motion. And this is not only size, what you can do, you can count because you know the volume, and you can see the potential. Particle metrics is coming from the field of the electrochemical characterization of particles, such as zeta potential measurement, which is related to the surface charge. And we found out this also can be done if you tag your particles with a fluorophore. You can actually do fluorescence, and this adds the benefit of specificity. What you can do with it? You can do quite a number of applications are here, basically nanoparticles. I will focus on the bio-nanoparticles, viruses, exosomes, liposomes. So this is a real-life video from 40 nanometer gold. Basically, they're rather busy in any kind of liquid, unless the viscosity is low enough, such as water. What then we are doing, because 100 years ago they were just looking at it and doing some quantification is rather difficult. So nowadays we use lasers, we use CCD or CMOS cameras, so we automatically detect and localize the particles, and an algorithm links it together for a trace. And basically the physicists like to talk about the mean display, the mean square displacement, which is basically the jump a particle makes from frame to frame. And if you do this for a lot of particles and for a long time, you can actually collect the diffusion coefficient for each single particle. So this is a single particle technique. And another famous person, two famous persons actually, Stokes and Einstein, they were helping us here in order to determine the particle size distribution and for the single particles, the diffusion coefficient. And if you know temperature and if you know the viscosity, then you're able to derive a particle radius. What else you need to know is it's a single particle technique. So if you're looking at only one single position with, let's say, 50 particles, you see something like that eventually. So you see something is going on, you measure maybe 50 or 100 particles depending on the optical setup. And if you do this for a large number of positions, as we are doing it automatically in our CW, you gain 1,000 particles within a minute. So this is rather quick because you need this in order to get reliable data, a reliable particle size distribution. So we can quickly acquire data from more than 1,000 particles, 1,000, 10,000 particles. It's done in an automated way, as it is presented here. We are moving the laser and the camera synchronously. This gives us representative data and valid analysis, and we have outlier control for each position, so we can easily detect good and bad quality of data. Yes, what about specific detection? This is a physical technique. It does not tell you what it is. It doesn't tell you if it's a bio-nanoparticle, it's a gold nanoparticle, so what can you do about it? Basically, you can tag your particles. So you have the particle and you can take an antibody which you have a fluorophore on it, and this gives you the opportunity to actually tag specifically your bio-nanoparticles. It can be exosomes, it can be virus particles, membranous, non-membranous particles, and you do a lot of fluorophores on the surface, and then if you excite it by means of the laser, you create the fluorescence light, and if you take a filter in place, then you filter out the scattering light from the laser, only capturing the fluorescent data. And the video here is shown from an HEK cell line derived exosomes with a specific antibody to a specific protein. And this works very nice, down to 40 nanometers, so we're still able to see down to 40 nanometer particles, meaning 10,000 fluorophores, but we can do better. These are basically depicting how it works. If you have fluorescent particles, meaning every particle is fluorescence, then of course in scattering and fluorescence mode, you should see the same concentration. This is true. And if you do a one-to-one mixture of fluorescent and non-fluorescent particle, you see half of it. So this is how it works. You can quantify ratios of fluorescent and versus non-fluorescent particles. So how good does it go? You can measure non-fluorescent particles, and in fluorescent mode, of course, you see 0 particles, whereas in the other case, you see 100% of the fluorescence particle. And the question was, how good can we do that? Because not all of the particles bear certain surface characteristics like proteins. So the question was, how far can we go in regarding the mixture of scattering and non-scattering particles? So we did this 101, so we can still see it. Then we increase concentration. So in scattering mode, it's getting quite busy. And we can go actually up to 10,000 to 1 ratio, and then the concentration of our standard was actually too low. So it's basically a lack of concentration of standard, and the instrument still can see it. So the take-home message here is we see one single fluorescent particle in the presence of 10,000 unlabeled ones. And we can do membrane staining. This is done here for typical EVs, and they are stained with CMO cell mask orange, and we excited 520 nanometers. This is not optimum, but we still can see it, and we detected 550. So basically, the CMO stain is going into the membrane, the plasma membrane, and so we can achieve a yield of about 85%, which is rather nice. If you do this specifically with an antibody, you expect a broad range of ratios, and as I have shown, our instrument can do up to 10,000 to 1. So even if you do not have a lot of surface or particle having the same surfaces, we were still able to see that. So there are a bunch of filters, bunch of fluorophores we are using, so this has proven to work. And we think it's a complementary technique for small particles. So combination of flow cytometry and NTA is a rather good thing to do, so you do not miss the small particles. What about surface charge? If you apply an electrical field by means of electrodes, the particles bearing an electrical charge will move in this field, and this is also known a long time in the field of colloidal chemistry, over 100 years ago. Basically, you're using the migration of the particles in an electrical field, and basically what we measure is the electrophoretic mobility. This is basically the velocity in an electrical field. And Helmholtz-Smoluchowski, more than 100 years ago, derived theories how to do that, how to derive the zeta potential. This is basically the energy you need in order to remove ions or charge from the surface of the particles, and this gives you idea about stability as well as particle, what is the charge and the sign. If you do titration, you can actually get the charge, the total charge of the particles. So this basically, it determines what kind of surface charge you have, whether negative or positive. So we did polymers, we did protein aggregates, exosomes. So depending on the surface charge, you will see this in the zeta potential. And it's a nice thing to do it, first measuring size, then measuring charge, but we actually can do it in one run. Basically, zeta potential and size in one measurement at the same sample. So we get scatter plots, which is quite nice in order to do quantitative data. And we also did lecithin liposomes, a pH titration, basically the isoelectric point for those liposomes, and you get nice 3D graphs, and the animation shows you the characteristics of the scatter plot upon different pH. Multidimensional analysis. So basically, we have a video, and a video contains a lot of information, and it would be a shame not to use it. So what can you do? This is a typical NTA image, and if you look closer at it, we have rather weak scattering particles, like polystyrene, and we have a rather bright scattering particles, such as gold. So we made an artificial mixture of gold and polystyrene, and basically based on the intensity, you can do additional, again, scattering plots, and you can get additional data out of it. We were wondering how complex can a mixture be? So this is a trimodal mixture. You already can see a separation of at least 2 peaks. But if you then do multivariate analysis on it, taking into account the intensity, for example, for the scattering plots, the algorithm can automatically detect subpopulations, and in this case it was an artificial mixture of gold liposomes and polystyrene, rather large ones. So taking into account additional information really helps you in identification and quantification of complex mixtures. Let me summarize it. So nanoparticle tracking analysis gives you size, gives you count, zeta potential fluorescence, plus with the multivariate statistics, you can really look into the data and can do some quantification of complex mixtures. And I'm at the end of my talk, and thank you very much for your attention. We actually have a booth out there, so we have much more application than what was in the talk. If you have questions, if you can do certain things, such as proteins on surface, titration and stuff, please come to our booth. We are ready.
Thank you. Okay, so if there's a burning question, you can ask Clemens right now. Please hold up your hand if you have something you want to add or share or ask. And if this is not the case,
we will welcome you in our booth. Thanks a lot for your attention.
Otherwise, at the end of the session, there will be more space for Q&A. So